Hi guys, the topic for today is molecular therapeutics for human inner ear diseases. Introduction. The last 30 years have seen an explosion in the understanding of the molecular basis of hearing loss. Currently, there are no pharmaceuticals designed specifically for the inner ear. Development of the therapies to treat hearing loss and balance disorders is complicated by the diversity of disease processes leading to functional loss. Well, the treatment of the hearing loss with the hearing aids has only a 30% penetration and a low rate of patient satisfaction. Inner ear is separated from the general circulation by a blood-cochlear barrier with similar properties as the blood-brain barrier, making systemic delivery of the medications less efficient. This is just a diagram showing you the blood supply to the inner ear. This is the ICA, the anterior inferior cerebral artery. Then this is labyrinthine artery, the common cochlear artery, the posterior cochlear artery, vestibulo-occular artery, cochlear branch of the vestibulo-occular artery, vestibular branch of the vestibulo-occular artery, and the anterior vestibular artery. So these are all the small arteries supplying the inner ear. We will make a separate video on the blood supply of the inner ear. So here you can see the inner ear is easily accessible via the round window membrane. So here you can see this is a round window and it has a membrane over it. So additionally, inner ear can also be accessed via cochleostomy through the Stapes foot plate, via a canalostomy, or through the endolymphatic sac. We can go through we can go into the cochlea while doing the cochleostomy. We can go into the inner ear by the endolymphatic sac. So there are various routes by which we can penetrate we can administer any vector or any drug into the inner ear. One of the significant advantages of the inner ear is that it is relatively closed space. So the delivery of a drug or a molecular therapeutic such as the viral vector or into this space would have significant advantages over the systemic delivery, in which the large doses would be needed in order to distribute the drug into the inner ear. This is so true. Yes, it is a closed space with a barrier, blood cochlear barrier. So yeah, now the development of the vector for the use in the inner ear. An ideal vector for the delivery of the genes to the inner ear needs to be specific and concentrated in a low volume to avoid the hydraulic trauma when delivering to the perilymph or the endolymph. So the dose should be very precise, as it may lead to hydraulic trauma into the inner ear. And the specificity can be conferred by designing a vector that contains either a tissue specific promoter or a surface component of the vector that ensures tissue specific binding. This would allow vector dose to be minimized and the gene of the interest to be expressed exclusively in the target cell. Ideal vector would also have a low incidence of the electric immune response from the target host. Obviously, the vector which we are using should have a very low immune response or negligible immune response into the target host. So several different types of vectors are available to the delivery of molecular therapeutic to the inner ear. They differ in the complexity of the construction, the expression time, the potential side effect, and carrying capacity. Picking the right vector for the different disease targets is a key factor in developing a molecular therapeutics. A variety of other viral and non-viral vectors have been tested in the inner ear. The most current studies are focusing on optimizing the adenovirus and the adeno-associated virus based vector system. So this virus has been selected, adenovirus and adeno-associated virus system. Now here you can see how this works. So this is the vector. This is as you can see, this is the adenovirus, the vector. So what happens? It binds to the cell membrane. Then the vector is packed in vesicle. Then the vesicle breaks down, releasing the vector, and the cell membrane protein makes the protein using new gene. So this is the DNA inside this vector. So this is the viral DNA, and this is the gene of which expression we need. So this gene goes inside this and gets incorporated, and it is a very specific gene for a specific part in the inner ear. Like you need to these gene to act and get incorporated into the supporting cells of the hair cells, lead to hair cells, inner hair cells regeneration. So these are very new specific genes. Now the adeno-associated virus is a small parvovirus that carries a genome flanked by a terminal repeated sequence. These sequences are needed for the initiation of DNA replication and the viral packaging. Native virus can exist in a lytic state, lytic life cycle, or can integrate into the host cell DNA as a provirus. A helper virus, adenovirus, is needed for the virus to complete the lytic cycle or for the production of the adeno-associated virus. This quality of latent state allows this vector to potentially be used for treatment that require long expression period, such as the correction of the fixed genetic deficit or production of growth factors that would be required over extended time periods. The life cycle of the adenovirus does not involve the integration into the host genome. Rather, they replicate as an episomal elements in the nucleus of the host cell, and consequently there is no risk of the insertional mutagenesis. To develop the molecular therapeutics for the hearing loss and balance disorder, we need to be able to accurately diagnose a given disease on a molecular level in the inner ear, since the multiple different genetic deficits can cause similar hearing loss. So we need to accurately judge what is the defect and at what point the defect is there, so we can act the vector to act on the specific gene. For example, a hearing loss can potentially be caused by loss of the hair cells, loss of the spiral ganglion, dysfunction of the striatum vascularis on an unidentified process, all of which may lead to similar standard audiograms. Since we are unable to biopsy the living inner ear, we have to rely on having an accurate clinical history in combination with the appropriate physiologic testing. Loss of the vestibular hair cells results in the changes in the vestibulo-occular reflex response. Bilateral vestibular hypofunction results in the permanent chronic balance dysfunction and inability to fix a target on the retina while moving. So complete recovery from the vestibular loss may only be possible through replacement of the missing vestibular sensory cells. Well, if you talk about the aminoglycosides, yeah, it causes the vestibulopathy. It represents a population where we know the underlying pathology, vestibular hair cells loss, making the use of the molecular agent to regenerate hair cells possible. Focusing on the hair cells loss as a disease process allow us to identify a target supporting cell, pick a gene of series of genes that can be tested to regenerate cells. Now here you can see how this aminoglycoside antibiotics acts on this, because it acts in this diagram. You can see a simplified schematic diagrammatic depicting the aminoglycoside induced hearing cell death in a mammalian inner ear. Well, the aminoglycoside exposure causes a reactive oxygen species. So here you can see this aminoglycoside causing a reactive oxygen species, stress kinases and other caspases. So you know which caspases lead to further apoptosis. So it forms the stress kinases and the caspases family of proteases to become activated in the hair cells, so which further lead to apoptosis. Similarly, this is also a diagram showing the same thing, that aminoglycoside antibiotic causing the mitochondrial dysfunction and further leading to damage in the hair cells. These are the inner hair cells. You can see these are fine here and these are damaged here, leading to hearing loss. So molecular strategies for the regeneration. Two main types of the molecular strategies have been involved. The key difference between the mammalian and the non-mammalian vertebrates is the capacity of the spontaneous hair cell regeneration. See, the non-mammalian vertebrates can functionally regenerate both the vestibular and the auditory system, but this doesn't happen in the mammals. So mammals, this capacity correlates with the neuroepithelium's capacity for mitosis. So in non-mammalian vertebrates, mitosis occurs in these supporting cells of the neuroepithelium, which has a capacity to regenerate, but in the mammals this property is not there. A potential approach to this problem is of the cell cycle control, allowing division of the supporting cells and the differentiation of these cells into the hair cells. So we know that these supporting cells in the inner ear, which further differentiate and forms the divide and differentiate and forms the hair cells. Well, the ablation of the retinoblastoma protein in the neonatal mice results in the proliferation of the supporting cells. Although the pharmacologic inhibitors of different components of the cell cycle are available, use of a gene therapy approach would allow individual cells to be targeted so that, for example, cell cycle control is the only interrupted in the supporting cells. A second approach is to take advantage of the observation that a non-mammalian hair cells can be generated from the supporting cells by transdifferentiation. This could potentially be achieved by the overexpression of the gene that control the hair cells genesis and altering the expression of gene controlling pattern of the neuroepithelium. So in the simplified way, you can see in the mammalian, in the mammals, what happens, this is the hair cell. Normal hair cells, their hair cell death is occurring. So there are two ways we can regenerate. One is by the mitosis and differentiation of the daughter cell. So one is this, and other is this direct, there is a conversion of the supporting cell into the hair cell. So in this there is a division is taking place, and here there is a direct conversion of a supporting cell into the hair cell. So what is the effective delivery of the vector to the inner ear? Several different approaches have been evaluated for the delivery of the gene therapy vectors into the inner ear in mammals. The most studies have used a cochleostomy or the injection through the round window membrane, or to deliver vector to the perilymphatic space using a direct injection or an osmotic pump. Cochlear function measured by the ABR showed minimal threshold shifts in the canostomy animals. So cochleostomy, however, resulted in the threshold shifts significantly higher than those seen in the canostomy group for all the three frequencies tested. So the author concluded that the canostomy provides a procedure for transducing both vestibular and the cochlear tissue, as well as the preventing the cochlear function. Alternate delivery modalities to the inner ear have been tried, including the delivery to the endolymphatic sac, delivery to the round window, as well as the delivery to the scalar media via the cochleostomy through the stria vascularis. Now, the identification of the necessary component of the successful molecular therapy. Gene must be efficiently delivered to the appropriate target cells. In the case of the ATOH1 supporting cells, in order for the gene to have desired therapeutic activity. The choice of vector system is one of the key to building a molecular therapy, since it determines whether the therapeutic gene is delivered appropriately. Adenovector have generally been shown to have a limited expression time, making them the ideal for the delivery of gene, where only a limited time period of delivery is needed, such as the ATOH1, which is active only during a brief time during development. One of the potential disadvantages of the adenovectors is that they can induce the inflammatory response and be recognized by the pre-existing immunity. This can change the clearance characteristic of the vector and also potentially result in the deleterious effects on the target tissue. So multiple studies in the different labs have shown the potential of the gene delivery, and in particular the ATOH1 transfection to produce the functional sensory hair cells in a variety of the model systems. In order to translate this observation into a clinically relevant molecular basis therapeutics, we need to focus on easily diagnosable diseases processes. Thank you.